Welcome to the last session, and it is my real pleasure to introduce to Sarah Teichmann, our second keynote speaker and professor of stem cell medicine at the Cambridge Stem Cell Institute. I had the great privilege to do a postdoc in Sarah's team and really witness firsthand how Sarah is a stellar scientist with highly creative ideas, while at the same time being one of the most generous persons I have ever met. And her lab has made seminal contributions to the field of genomics and bioinformatics. And for this, she has received numerous awards, including the EMBO Gold Medal and also the Biochemical Society Award. And also, she is an EMBO member and a fellow of both the Academy of Medical Science and the Royal Society. So her lab is at the interface between computational and experimental molecular biology, and she has pioneered highly used tools to extract biological information from the single-cell genomics datasets. And really, using all these single-cell genomics tools, she's redefining our understanding of human physiology, including transformative insights of the immune system that I'm sure she will be talking about. And her team also has made major contributions in defining the biophysical mechanisms in protein complex assembly. And really, this deep knowledge in proteins, molecular and computational biology supercharged the lab's creativity and outputs. And I particularly benefited from all of this when we developed CellPhoneDB together. So also Sarah is also a true scientist leader. She's co-founder and co-leader of the International Human Cell Atlas Consortium that aims to map all cells in the human body in 3D, and as she mentions in her talk, also in 4D now. So please join me in welcoming Sarah Teichmann to the stage.
Thank you, Rosa, for that kind introduction. That's really humbling, kind of to be introduced in such a kind way. And thank you to all the organizers for having me here and putting me in this session so that I could make it from Stockholm. It's really fantastic to see the community thriving and growing, and that so many of you have come here from all different places in the world, and also that we now, since the pandemic, have the hybrid option. So there are people online, and a very warm welcome to you as well. So without further ado, I think, is this, let's get the slides up and running. It's the, sorry folks, hang on a minute. Here we go. There's something funny about which display it's showing. Now into 3 and 4 dimensions, and Alex's talk kind of showed that so nicely. So what I'd like to focus on in this talk is really the human, so ourselves, our own system, and where the insights that we can get at this juncture, sort of at this particular point in time, where we have draft suspension cell data from many, many tissues in the body and are now sort of putting Humpty Dumpty together again, kind of in terms of not only the tissue scale, but also actually the organ scale and the whole body scale. So if we sort of look back at quotes, Faye had a nice quote from Eric Lander. I'm going to go even further back in time to Sidney Brenner, who over 20 years ago in his Nobel lecture said, we need a program of making maps of cells and maps of how cells talk to each other. The cell map project, for which we don't need a model organism, so in other words, we can study ourselves, kind of the human body, will be one of the things to occupy us for the next few decades. And really, then sort of he continued with his obsession of cell maps, which obviously his, the C. elegans, the worm lineage tracing that he and John Solson did was the model organism cell map in some sense, if you like. And so continuing on this idea, after the completion of the Human Genome Project, he said, we now have an unprecedented means of collecting data. However, it's the understanding that's lagging, and we need the correct level of abstraction to interpret the genetic code. And one level of abstraction is that's very powerful is the cell, because it's the fundamental unit of structure, function, and organization of living systems. So the cell map is at once a map of the molecules within the cells and a map of the cells within the organism. So it's this molecular layer, that's so informative. And so in a way, going from the Human Genome Project, to single-cell genomics was giving us that way of interpreting the genetic code, because the cells are essentially, like we've heard from Alex and others, they're interpreting the gene regulatory modules, basically reading out the genetic code through the transcription factors that are recognizing DNA in a sequence-specific way. And so the cell map, or the Human Cell Atlas project, is a way of deciphering that code in that sense as well. And it was the resolution revolution in genomics, the single-cell genomics revolution, that really drove this whole era, and of course this community, and why we're here in this meeting today. And that started off just down the road from here in Azim Sarani's lab in the Gordon Institute, when Fuchu Tang and Azim and their collaborators were mapping a handful of cells that were handpicked from the early mouse embryo, because basically there were so few of the cells that they were interested in, so they had to handpick them and study them one at a time. What was clear to many of us, and many of us are here, including Itai and others, was that would open up then the possibility for also understanding cell compartments, cell populations, tissues, and so on, if you could scale up that technology, right? Immediately it kind of opened up a whole sort of plethora of opportunities, and that's been an incredibly exciting journey. And then hand in hand, we've had the spatial transcriptomics technology development along the sort of imaging and the sequencing axis, and we've had some beautiful talks here about this. So if we want to, assemble the cell atlas of the human body, then the challenge that we're faced is obviously that these technologies are amazing and developing apace. But the human body is of a scale, on the meter scale, basically, that where we're faced with the challenge of linking the molecular, that molecular map within cells and the sort of cell-cell communication with the whole organ and body structure scale. And for an embryo, you can do this. This is a recent limb development paper from us with Hongbo Sheng, who's another alumnus, and collaborators, Barbara Wold and others, where this is a stitching together, where we're sort of tiling the limb with this Visium stitcher approach from Nadav in the group. And in that way, you can kind of put together the pieces to get a continuous structure. But that's obviously it's possible for an embryonic tissue or, let's say, a mouse, but it's not possible for an entire human adult. And so that's sort of the challenge that we're still faced with. And of course, the year that we had the first incarnation of this meeting, 2016, so Rosa said that March 2016 was the first version of this meeting. So that was still when I was a joint faculty between EBI and Sanger. And we launched this series with the Connecting Science staff. And in October, we launched the Human Cell with a meeting in Wellcome that we co-organized with Aviv Regev, who was then at the Broad Institute, and others, and together founded the International Human Cell Consortium, which has this aim of making that comprehensive reference map. And I should say to everybody, this is an open, grassroots, scientist-led community. And if you haven't joined yet, please sign up if you're interested in this mission of mapping the human body. And where we stand now is that as a community globally, with over 3,000 members, we've mapped over 100 million cells. And you can see the different organs and systems that these come from, and the different sort of biological networks or interest groups that work together to achieve this. And so this is really a nice, a nice achievement over, I guess, 8 years or so, and especially taking into account the pandemic, which kind of was the scenic route on this journey, kind of through viral infection and immunology and so on. It was challenging, though, to kind of achieve or push further the human cell as a healthy reference map, as it were, but it gave us a lot of insights at other levels. So what I want to focus on today is the concept that the cell map basically tells us about tissue architecture and tissue structure, and at the same time function, because a lot of the function of a cell is actually informed through the niche and the cell interactions that it sits in, and we've sort of heard a lot about that today. And I want to give a short introduction into the first organ that we mapped, which was the placenta, and that's of course driven by Rosa and collaborators. And then in this case, what we're learning through the niches is how fetal tolerance is achieved by through the crosstalk between the maternal immune system and the fetal cells that, of course, harbor the paternal antigens. And then the heart, which is an incredibly complex organ with 30 or so different tissue niches, many different tissues. It's not quite as complex as the brain that has over 100, but it's a really, there are different immune niches, there's crosstalk with the endocrine system, the nervous system. Of course, it has its own kind of conduction system, the electrical conduction, the cardiac conduction system that's like the brain of the heart to keep everything coordinated. And so how do we connect all these things to understand the function of the organ at the molecular and cellular level with these new technologies? And then finally, I want to go on to our efforts in mapping the thymus as an entire organ and also throughout development and postnatal stages. So starting off with the placenta, which may have been introduced earlier, we have the sort of conundrum that we're faced with, and this is one of Rosa's beautiful slides showing the kind of key interface where the maternal immune system meets paternal antigens. And so that's a long-standing conundrum in immunology because obviously in the thymus, which we'll hear about later, with T cells are trained to tolerate our self-antigens, but basically meet challenges from non-self. And how is it that then the T cells at the in the womb, basically in the decidua, which is the pregnant form of the endometrium, how is it that they're able to tolerate the antigens that are coming paternally? And of course, 50% of the alleles in the fetal cells on the placental side are paternal, so non-self, if you like. And there are various different pieces of evidence in the literature that kind of explain that, and they range from special HLA to special fibroblasts on the maternal side, special HLA on the fetal side to special fibroblasts on the maternal side to regulatory T cells, and it was really the regulatory T cell that was what triggered in my mind the curiosity, because we were looking at mouse T helper cell compartments, so CD4 T cells, which can be effector and sort of responsive immune cells on the one hand, but also are really interesting in that regulatory T cells on the other hand are kind of down-regulating our inflammation. And so we were curious to understand how all of these compartments work together to tolerate the paternal antigens as the fetal cells, which are shown in red, enter the maternal tissue, and on the one hand lead to the reshaping of the blood supply in the artery, but on the other hand they have to kind of stop at the interface of the myometrium. So when the endometrium finishes and the muscular outer layer sort of starts. And so what Roser did in those early days, and I remember also Jong-un, who's here helping, was using SmartSeq to tackle each of the cell compartments one by one, and that was pretty crazy effort. And then we were very lucky in that the droplet-based techniques came along, and the nice 10x robot made everything easier. And so then, sort of through the combined data, basically from these different compartments, we're able to cover the different stromal compartments. And you can see here from the histological image, very clearly different morphological regions that are defined also by different fibroblast populations that have interesting functions. The myometrium is down here. And these different zones are kind of like different zones of our skin with different, but in this case, both different stromal and different epithelial compartments that we're able to chart at the molecular level for the first time and define at full molecular breadth with these technologies, and understand all the different layers and what how they operate, and also how the fetal trophoblast cells interact with the different NK cell subpopulations that we're able to define in terms of different activation states and different cell surface receptors and their interactions, on the one hand with the special HLA, and on the other hand with the maternal stromal and macrophage cells. And what you can see here is all the different receptor-ligand interactions involved in this crosstalk. And as Roser said, I sort of had a previous life in protein biophysics and protein assembly. And that, a couple of years ago, there was a Nobel Symposium on essentially protein folding and the AlphaFold era and so on, that was organized by Arne Elofsson, and that community knows me from this more protein structure and assembly work, where we were studying the subunit architecture of protein complexes, how they assemble once they are synthesized by the ribosome and need to meet their friends inside the cell. And of course, then some of those proteins are displayed on the cell surface and engage in receptor-ligand interactions. And it was that awareness of subunit stoichiometry for the receptor-ligand interactions that was the inspiration for cell phone. And as Roser said, Roser kind of picked up the glove, picked up the challenge together with Mirjana Efremova, and integrated not only the complexes of known three-dimensional structure that are responsible for the cell-cell communication, but also from a lot of biochemical and genetic and so on databases. And then earlier this week, we had this Nobel Symposium on Monday and Tuesday, so yesterday and the day before, and yesterday afternoon, I was giving this seminar, and that's why, and Faye was speaking on Monday in Stockholm, and that's why we had to miss the earlier days of this conference, which was a real shame. But we need more cell-cell communication across the different meeting organizers, kind of, so to avoid that. But using this cell phone, we're able to then map the communication between the different immune subsets, the trophoblast compartment, the stromal and the epithelial cells on the maternal side. And Roser then took this forwards also to understand the whole menstrual cycle, which is kind of the precursor to this early stage of pregnancy, first trimester pregnancy. So you have this very dynamic tissue in the uterus where you've got different stages, and then obviously menstruation kind of and destruction of the inner layer of the uterus at the end. And this was really a tour de force that Roser took on with Luth, Garcia, Alonso, and others at the start of her group over the past few years. And I want to give a big shout out to that work, which is also the foundation for informing in vitro systems mimicking the human endometrium, which is so different from mouse, and which is why it's so important to study the human. So as Sidney Brenner said, we need to study ourselves because we're different from our furry friends who don't menstruate, for instance. And this has just been covered by The Economist, how the human cell atlas data can inform the organoid systems and provide ways that we can study this system, for the first time in a comprehensive human-focused way, which is really important for women's health, for development and successful development in terms of the placenta and the embryo. So this was really the first human organ that was mapped across all tissues and cell lineages, in the sense that Roser took biopsies from all the different parts of the placenta, so all the different tissues that constitute the placenta, the interior, the outside, and so on. And we were also able to cover all the different compartments, the trophoblast, the immune, the stromal, the epithelial, and so on, and define the tissue architecture by integrating the kind of old school single cell data with, which was SmartSeq and 10x and so on for the different compartments, with low throughput spatial transcriptomics, which was that immunohistochemistry, the single molecule FISH, and so on, for placing the cells in the different tissue niches on both the fetal side and the maternal side, and then infer how the cells are talking to each other. So we're kind of like doing a puzzle, basically, to put all the cells together without having the direct sequencing readout or the direct imaging readout of all the molecules in all the tissues. And so that was kind of the old way of reconstructing cell communication and cell function. And in more recent times, obviously, we've had these fantastic direct spatial technologies that allow this direct mapping of tissue niches and cell interactions. And with Carlos and Mona, who are actually here and who were just credited in the previous talk, they each have their own groups now in Würzburg in Germany. We started off mapping the heart with collaborators in Berlin, Norbert Hübner, and in Imperial College, and in the US. And then we went on more recently with James and Kazumasa, who I think aren't here, to expand the different tissues. So not just look at the muscular free walls of the four chambers of the heart, the atria and the ventricles, but also study the cardiac conduction system. And that's much more challenge. It's a challenge because catching kind of the rare cells like the pacemakers in the cardiac conduction system is really tricky because they're such a tiny number, and they're sort of hidden and not visible to the eye as to where they are in the heart, and actually their location in human shifts around slightly. So it's not possible to just surgically resect the sinoatrial node where these pacemakers sit. The pacemaker cells are amongst the only spontaneously firing cells in our body. They start beating early in development and are then responsible for our heart rate, and continuously fire many dozens of times per minute until we sort of take our last breath, and that's the end. And it's this spontaneous firing that's responsible for obviously delivering oxygen and nutrients around our body through the pumping of the blood. And it's modulated by various factors, including input from the autonomic nervous system and also endocrine factors. And what was unknown was the molecular profile of pacemaker cells in human. There had been mouse data. Again, the mouse has a heart that's much smaller. It beats much, much faster and has different features to the heart, as I'll show later. And in terms of the connectivity in this niche, what's important is understanding how those cells are served in a way by in their niche, and how they're electrically insulated, and how they're innervated by the sympathetic and parasympathetic nervous system, which is increasing the speed and decreasing the speed kind of the pacemakers. So in this recent work kind of published at the end of last year, we had almost 750,000 cells and nuclei and categorized into 12 coarse-grained cell types that we'd also defined before, but 75 fine-grained cell states. And here we had not just the single nuclei RNA plus attack sequencing for gene regulatory network inference, but also the direct readout of the spatial microenvironments with the 50-micron Visium spatial transcriptomics chips. And so for discovering spatial microenvironments here, we took two different parallel tracks. One was taking the manual structural annotations with our collaborator Yen Ho, who's a cardiac microanatomist and pathologist at Imperial College. And you can see her manual annotations here. So here is the myocardium, which is the muscular regions of the heart with the cardiomyocytes. There are regions of fat. You can see little regions of fibrosis here that she's annotated with her kind of inspection of the bright field microscopy image of the H&E stained section. And this orange thing is the sinoatrial node. And that's what's so hard to see for the surgeon, or for surgical, when you're surgically dissecting the heart, because it's hard to, it's a microanatomical structure, and it's not entirely stereotypically located. Can vary slightly from one human heart to another human heart. So we took her structural annotations and looked for enrichments of cells in her annotations, and then we also used unbiased non-negative matrix factorization. So looking for patterns of cells that are co-occurring after deconvolution with cell to location, with a reference of the single cell and single nuclear data that we had. So it's a sort of, again, like a combined detective work to identify co-occurrence of cells and which tissue niches. And we harmonized the two approaches with each other to identify niches, and then further developed cell phone building on the receptor ligands that were quite immune focused and purely protein focused, to also have G protein coupled receptors with small molecule, also with small molecule ligands that we can trace their presence. Of course, they're not genes, but the enzymes that synthesize the small molecules and the transporters that transport out the small molecules for the receptors, they are encoded by genes, and we can trace them by their expression patterns. And so that sort of add-on module was developed for the cardiac conduction system. And using these two ways of defining spatial microenvironments, we're able to see, kind of quote-unquote, a difference between the center of the sinoatrial node, where the pacemaker cells are sitting, those are these cells here, and the periphery of the node, where there are macrophages, fibroblasts, and adipocytes and neural cells. So the real excitement for the cardiac anatomist like Yen and the pathologist is that the genomics and data science methods allow us to sort of see things that the microscopy from the pathologist wasn't able to see before. And of course, so that's a very exciting kind of time from that point of view in terms of redefining pathology with genomics and data science. And the niches that popped out here, in addition to the sinoatrial node and these subcompartments of center and periphery, are the epicardium, which has immune niches that harbor IgG on the one hand and IgA on the other hand, sort of two separate niches for plasma B cells, and these cells are co-occurring and cross-talking with myeloid cells and fibroblasts that give them both chemokine input but also BAFF-APRIL signaling to stimulate the B cells in a non-germinal center mode of communication close to lymphatic vessels. And this is in the epicardium. The epicardium is the outside of the heart, and basically we think those immune niches are protecting our heart from the dirty lung that's next door. And then what we're also able to detect are these micro fibrotic regions that I showed you were manually annotated, and they have families of cells that again include specific myeloid populations that are giving input to fibroblasts by TGF-beta, OSM, and BMPs, and activate them to lay down collagen. And so this is obviously not a great process because it's stiffening the heart through this deposition of extracellular matrix. But it is occurring with aging all the time, kind of as we get older, and it's in these hearts that I should emphasize are healthy, quote unquote. So they're declined transplant donor hearts, and this is a natural kind of process that's occurring as we age. They're not diseased hearts. So I've mentioned the pacemaker cells. They're here in the sinoatrial node, and they're spontaneously firing and sending those signals to the cardiac conduction system via the Purkinje fibers and the Purkinje cells that then basically tell the ventricles and the atria to contract in a coordinated way. And obviously, if that goes wrong, then there's things like atrial fibrillation disease. So it's very important to have that conduction and the pace kind of being correct. And in the pandemic, collecting these declined transplant donor hearts was really challenging because in the UK, transplantation went right down during the pandemic. And James and Casamassa kind of traveled around the UK as much as they could to Newcastle and to London, working with Michaela Noceda's group. And from the first 2 transplant donor hearts that they dissected, basically we had about 50 nuclei that had high expression of this calcium channel, CACNA1D. And that calcium channel is in the mouse, is Mark's pacemaker cells. And you can see that this sodium channel is high in these other cardiomyocytes, which form the bulk of the atrial cardiomyocytes and are typical of working cardiomyocytes. So that led us to suspect that these are indeed pacemaker cells. And in the group meeting where James showed this, I thought, wow, is this really true? And then what they did, James and Casamassa, was mapping all the ion channels and receptors, and with GPCRDB and cell phone and so on, comparing the neural cells in the heart to the atrial and ventricular cardiomyocytes and the pacemaker cells. And you can see that the pattern of the pacemaker cells here is very different from the other cell compartments. And then when we map that pattern back onto the spatial transcriptomics, they're indeed in the center of the nodal region, as I showed you before. And they're co-occurring with glial cells, actually with neural cells, and with particular fibroblasts in this niche. And then when we go back and do validations with immunohistochemistry, we can see that the purple pacemaker cells are kind of hugged by these glial cells that are nourishing them. And we show that they're interacting by glutamatergic signaling, basically, which is unexpected, but we trace the glutamatergic, the glutamate biosynthesis and the transport and the receptors across the cells, as well as the acetylcholine signaling that's known between the pacemakers and the sympathetic and parasympathetic nervous system. And what we can also reconstruct from the expression patterns is fibroblasts that are secreting particular extracellular matrix or synthesizing particular extracellular matrix proteins to constitute the entire niche in that region. Now, as I said, the beating rate of the heart is really crucial, and there are a lot of drugs that control that are so-called chronotropic and that control the rate by either increasing or decreasing it. And so that led us to think, well, we can again work with our database friends at the EBI and use the Kemble database. Sorry, I thought I'd marked it. Maybe I didn't. But the point is that we go to the Kemble database, which is the open source database of all drugs and compounds in development and the known targets that they have in human and which phase of clinical trials they're in, what are the side effects and so on. And we can then map them onto the gene expression patterns from the single cell data of the different cardiac cell types, including the pacemakers, with our drug-to-cell computational statistical pipeline. And what drug-to-cell showed was that the GLP-1 receptor agonists like liraglutide and semaglutide, which are mimicking GLP-1, which is the pre-processed form of glucagon that's made by our pancreas and is signaling to the brain whether we're hungry or full or sated, basically that is predicted to bind to a receptor, the GLP-1 receptor, within the pacemaker cells. And that was kind of unexpected and unknown previously, because why would a hunger hormone, synthesized in the pancreas and the gut, why would that be acting on the heart? And just sort of for clarity and context, the drugs are liraglutide and semaglutide. So Wegovy and Ozempic, which are those blockbuster best-selling drugs for diabetes and obesity, from Novo Nordisk and Eli and so on. And so the surprise here is that we can infer cell-cell communication, that we came across through this drug mapping exercise, not only in the niche, but also from faraway locations in the body, and kind of have the cell phone, if you like, is not just paracrine, but it also can be used in an endocrine sense. And what this suggests is that GLP-1 analogs are acting via direct action on pacemaker cells, and this is and not by some indirect mechanism. And this is backed up by the sort of epidemiological evidence from the clinical trials that there's a 6 beats per minute increase in heart rate in people who take these drugs. That's obviously offset by the cardiovascular benefit of the weight loss of the drugs, but it's it suggests that from the clinical data that there's an effect. And when we culture the fetal human cardiomyocytes in vitro and add the drug, we see in vitro that there's a change in the beating rate. So there's both in vitro evidence and the clinical evidence that there's this direct mechanism of action that we predict from the drug-to-cell and cell phone kind of human cell atlas type of approaches. So the summary here is that the human cell atlas data is a molecular guidebook for drugs, for of course also for many other applications like genetic variants, like viral entry factors, the way we did in the pandemic, for endocrine signaling from distant places in the body. And we can match drugs to cells and cells to drugs, and this obviously has implications for toxicology and repurposing. So to summarize the niches in the heart, we find these immune reservoirs in the epicardium that I said is sort of protecting the heart, if you like, with the IgG and IgA B cell plasma cell compartments. We can assemble basically through the different tissues, the pacemaker cells, the Purkinje fibers, the Purkinje cells, the whole cardiac conduction system, by taking the 8 different regions that we studied in this project and link it to the autonomic nervous system and also to the endocrine system via drug-to-cell, the distant signaling. So again, I just want to say very quickly thank you to the DREAM team that worked super hard in the pandemic to collect and analyze the samples. And James and Kazumasa. James is a cardiology clinician scientist PhD student who's defending his thesis now and is back in clinical training. still doing part-time research with us. Kazumasa is an incredibly talented Japanese postdoctoral fellow who's continuing in the lab, and Carlos and Mona are obviously the ancestors kind of this project in collaboration with the Hübner lab from Berlin and the Michaela Noceda's lab at Imperial College. And I just want to emphasize again that all of these projects owe a huge debt of gratitude to the donors and their families, without whom this wouldn't be possible. And thank you to the funders. In the last few minutes, I'm going to go on to talk about the thymus, and I've mentioned earlier that this is the site of T-cell synthesis, and it's also a little organ that's stuck next to the heart, and you may not have heard of it unless you heard Tom Tagan's talk earlier, or from his group members, who I know many of them are here, and they are long-term collaborators from Belgium on this work, and also Jong-un, who started this in the lab, who's here. And it's a fascinating little organ that was for many years just thought to be a piece of fat next to the heart, and then in the 1960s it was discovered by Jacques Miller in London as the site of T-cell development, so actually to have immunological function. And what happens is that hematopoietic stem cells, which are synthesized in the liver during pregnancy, and in the bone marrow in later development and after birth, and of course in our bodies as adults, find this organ by migrating, traveling through the circulation, and then get signals, input signals, notch signaling, and start to then kick the cells down a thymocyte T-cell differentiation route. And then the mature T-cells that are outputted, whether they are helper, killer, regulatory, and so on, they then travel around the body and colonize our tissues, and presumably have roles in organogenesis, and during development, in fetal development, but then also in defense, obviously, across all the tissues. And now, of course, we also know that they have therapeutic potential. So this is a relatively small organ, kind of on much smaller than the heart on the centimeter scale, and it's endoderm derived. And the question that we're then asking is, can we map the whole organ by modeling multiple measurement modalities together? And can we? How can we know where a particular sample is located? How can we know where tissue section is from? Because you're obviously limited to either you have the cells in suspension, which really don't have any dimensionality, they're just kind of suspended, or you have a tissue section which is like a 2D salami slice, and we don't really know where it's oriented. So how can we put all these pieces together into a kind of two or getting to three-dimensional map? And here you can see like a 2D stitching here with the Visium stitcher for the limb, but in the thymus, what we had was not just Visium, but also Ibex, because we had fantastic collaboration with Ron Germain's group in this Thymus Atlas Consortium supported by CZI, and also one-shot kind of image acquisition with a RareSite commercial platform with Omar Bayraktar's group, then many single-cell and single-nuclear RNA modalities, and CiteSeq with a 143-plex protein panel with Tom Tagan's group. And we've got different stages of development, from embryonic, so first trimester, into fetal, second trimester, and then sort of postnatal, early postnatal and pediatric tissues. And that's what we're focusing on in this project. And then different modalities in terms of also antigen receptor sequencing, and you can see the different proportions of cells. So how do we put all these? How do we put all this data together? Well, one way of doing it is to model the organ that has a cortex, which is the outer layer, and a medulla, which is the inner layer, with a landmark-based approach as a single axis, sort of going from the outside to the inside in a two-dimensional sense. And we use landmarks like the edge, the capsule, and then the corticomedullary junction, which has a different texture but also different molecular markers, and then inside into the medulla. And we have kind of nonlinear distances basically between each of the landmarks, which gives us a scaling from the outside to the inside. And this was a great collaboration with John Marioni's group and with a joint postdoc, Nadav Yayon, who developed this, and also with Vishnu Ullman, who took over after John left. And that framework we call organ axis, basically to have a two-dimensional nonlinear continuum, so a continuous, where we can then map the individual modalities and tell us where we are in the corticomedullary axis, and then model T-cell, not just T-cell development, but also other features of the organ onto this axis. And one of the things, as the hematopoietic stem cells go through their different developmental stages, double negative, double positive, and so on, as they migrate through the cortex and the medulla, you can sort of think of them seeing different signals in the different tissue niches that are secreted by stromal cells, epithelial cells, immune cells, and even vascular cells as they're migrating. And the signal, the input that they get is from chemokines, from cytokines, from growth factors like ones that are shown here. And we can then plot basically on the different layers, like the different layers of the onion kind of thing, from the cortex to the medulla, the different strengths of signaling that they're getting, and compare, let's say, CCL25, so chemokine input in the fetal stages, which is in blue, to how similar that is to pediatric, to the fully mature organ. You can see there are differences here in the inputs of these chemokines, CCL25 and CCL19, in fetal versus pediatric, for instance, in the different stages, the different layers of the organ. We can also look at this from a cell point of view and say, okay, in the fetus versus the pediatric thymus, what's the population abundance of cell states at these different maturation phases in the cortex, which you can see here, versus the medulla, sort of in these lower stages. And you can see, for instance, the progenitors are more cortical actually during in postnatal thymus. A couple of other findings that we made on this fantastic sort of project together was the organization of epithelial cells in the medulla around Hassel's corpuscles, and we found that these little structures, which are actually human-specific, so they don't really exist in the mouse thymus in the same sense, they're epithelial cells that are dying, but also expressing very large amounts of keratins, and those keratins are mimicking our skin, and they're also mimicking mucosal interfaces. And that's kind of part of this feature of the thymus that I mentioned, where it's educating in the medulla, in the interior of the organ. This little, these cells are kind of training the T cells to tolerate our own antigens, so the skin, the gut, so it's mimicking all the organs in the body in terms of protein expression and training to tolerate ourselves, but then still respond to non-self. And the Hassel's corpuscles in human, which are these structures that increase as we age or in puberty. The thymus kind of involutes in puberty, but this increases after birth. These structures, they are basically in human associated with skin and mucosal interfaces. Other findings were sort of stem cell niches that we discovered in two different locations, in the cortex and the medulla, with two different identities, and also the decision-making between killer T cells, the CD8 T cells, and the helper T cells, the CD4 T cells, at the very early stages in the cortex. And the CD4 T cells basically being dragged into the medulla by this chemokine kind of gradients faster than the CD8 T cells, because there's a slightly different expression of the chemokine receptors in the two subsets as they part ways, as it were, from the precursor states. And so I've mentioned Nadav and Veronica. I also want to give a massive shout out to Lena, who's here from Tom Tagan's lab, who worked with them. It's been like an incredible journey in many senses, and also, you know, really fantastic international collaboration with colleagues at NIH, the Germain lab and Notarangelo lab, in particular Andrea, and Mus Hanifa's lab in Cambridge here at Sanger, the teams basically in cellular genetics, and also the tissue resources from human developmental biology resource, and the pediatric hospitals for postnatal thymus, the Freeman Hospital, Great Ormond Street. And I want to emphasize that the organ axis software is not just specific for the thymus. We've found that it's actually useful for many different tissues in the body, because this layered axis kind of approach, you know, can be applied quite generally, and even to the heart, to niches in the gut, and so on to other tissues. So it's quite a general concept. And the software is on GitHub, and the paper I showed on bioRxiv and is in press in Nature and will come out as part of the Human Cell Atlas publication bundle in late September, early October. And the scales and here are really where we're trying to go from the molecular to the whole body via the cellular and tissue scale. And the journey of the T cells, of course, continues outside the thymus to their mature locations in both lymphoid and non-lymphoid tissues. And I've mentioned earlier that you know, mechanistically we need to have in vitro systems to study these processes. In the thymus, there's the conventional T cell maturation, where the training to recognize self from non-self comes from epithelial cells, but there's also unconventional pathways via TT cross-presentation, where the thymocytes are educating themselves. And if we look at artificial thymic organoids and compare the T cell maturation in the dish to the T cell maturation in the thymus, then what we see is that in the dish, the artificial thymic organoids that Denithi and Chenchu and Emma compared to the in vivo thymus, they're recapitulating the TT, the unconventional T cell development trajectory. And so we can sort of predict the regulators, both using the extrinsic signaling from cell phone and the intrinsic programs using genes to genes. That's an alignment, a continuous gene alignment approach that's in press at Nature Methods, that's on bioRxiv, in order to identify missing factors that we need to add to the artificial thymic organoid system to mature the cells. And what Denithi and Chenchu found here was that, for instance, if we add TNF-alpha that's low in the organoids but high in vivo, then we get a type 1 innate T cell. We can then identify transcription factors that are still need to be engineered in or added to make the full killer T cell identity. And so that's just to kind of finish off that the Thymus Cell Atlas also provides a molecular blueprint for engineering artificial thymic organoids, you know, as in the endometrial example that Rosa kind of pioneered. And the lab has worked together on this work in again, a dream team combination of Chenchu Suo, who's a clinical lecturer in pediatric immunology, finished her PhD a while ago, Emma Dan, who's an amazing PhD student who's now a postdoc in Stanford, and Denithi Sumanawera, who's a Schmidt Accelerate Fellow in the lab and an incredibly talented computer scientist, other members of the lab, past and present, Maz's lab, who are our partners in crime on all the immune development work, and Mena's lab, and also John, who was at EBI. And so to summarize, basically, what I've gone through very quickly is that the mapping of cell types and cell states in their tissue niches tells us about the function of the cells through their molecular interactions in the different niches. And the examples have gone sort of from the kind of key tissues in development where we tolerate foreign antigens to the training of the T cells, and then to the immune niches, but also other systems. And so it's really the mapping of the body is driven by the genomics technologies, but also the data science, the connection to in vitro systems, and it's linking to medicine more and more now and transforming medicine. So I want to finish with my disclosures and say that we are having a big impact as a community overall on pharma and biotech, as well as genomics and computational technologies. This is really having an impact on real life and making a big difference. So all the discoveries that we're making about the human body, together as this community, is actually transforming precision medicine, if you like. I want to also highlight that if you're keen on the cell map, the Human Cell Atlas, come to Milan in September. There's going to be a really fun meeting at the Human Technopole, which is this cool new institute dedicated to human biology in Italy, and we're going to have a bunch of different topics, which will be a lot of fun. I want to say thank you again for the opportunity to speak here, and I also want to say for anybody in the Teichmann lab or any alumni who are here, please let's get together afterwards outside, and we'll take a photo together and catch up. So thank you again, everybody, for bearing with me, and thanks for the opportunity.
Thank you so much for the fantastic talk showing the evolution of the Human Cell Atlas. Questions for Sarah?
Thanks, Sarah. Very inspiring. I wanted to ask about the last story about the thymus. So I think one of the kind of heated topics in the field for a long time has been how are antigens presented to the T cells spatially across the thymus? Is it all the cells presenting all the antigens, some of the cells, specialized niches?
Yeah.
How did your data kind of, what's your take on this based on your data?
Yeah, so this is fantastic. So I didn't go into the thymic epithelial cells in a lot of detail. In this early paper, we talk about different subcompartments, which we've now refined as well. And what we've done, I talked a little bit about the skin in the Hassel's corpuscles and the mucosal interfaces and the mucins and keratins that are expressed by those cells. And then there are different subsets that are mimicking, the myotex, the neurotex, which have been known for a long time. In the mouse, there's a lot of work on the different subsets. The human have some similarities, but some differences to the mouse subsets in terms of the patterns. So certainly there are well-defined populations that we can chart. But it's not like there's one cell for all the different, one tech mimicking each of the 1,000 different states in our body, if that makes sense.
Yeah, I was wondering if you could estimate how much of the thymus a T cell needs to traverse to kind of get
a difficult question. Yeah, that's a very interesting question, and it's a mystery, and certainly it's imperfect. So some cells will escape without being properly trained. And so the immune system has a lot of, I think, compensating mechanisms, including the Tregs, which are getting happy to see them. They're happy to see us, right? They're happy to see our own antigens, and they are sort of then suppressing. So it's an imperfect system that has checks and balances, and it obviously goes wrong sometimes in autoimmune scenarios. Yeah.
Thank you. More questions?
Fantastic overview, of the whole story. To see it like this, it's really inspiring. One thing that scares me a lot from your papers, and just the many people, doing, really taking this challenge to their heart, is these rare populations of cells that require really profiling a lot of cells, finding them in the right region.
Yeah.
And for the large part, we've known about cells that exist in the body through pathology, disease, kind of like historical reasons, medical, knowledge that we have. Yeah. Do you think we, are missing a lot of these rare cell populations based on the approaches that we're doing? Ido Amit had this beautiful story on the cells that produce EPO, right? A year ago or something like this. I'm just like, do we need to provide, how are we going to find these rare cells? And how many rare cells are we finding by these approaches? And do you think we need to change the way we think about creating these atlases?
I mean, so in small structures like the embryo, you can comprehensively, let's say, salami slice the whole thing, right, and afford to just cover everything. In larger volumes, then I think taking an informed approach, like the brain cell community has done in dissection, or the way we've done here with the cardiac, like that's the only way to sample in an informed way, and then hope to catch the different structures. And you're right, there might still be some that are missing. I think there's a combination of the kind of like technical gridding, like a comprehensive gridding, with the anatomically informed, biologically informed kind of approach, and then together we hope to get to a comprehensive map. But I do think the comprehensive map, it is important, because also from a medical drug development point of view, you really want to have that comprehensive map for side effect avoidance and precision medicine development and so on. So it is important that we catch all the populations. I think it's a good point, and so thanks for that.
Yeah, hi Sarah, great talk. I mean, this is a really outstanding work. I was wondering, because you have this background also in the protein, from the protein side, and you were using this together with Rosera, I guess, for the cell phone interactions. I was wondering, where do you think we are with these cell-cell interactions? Do we know all the receptor-ligand interactions, or are we constantly discovering new ones? Now also with your new module that you added with the GPCRs, is there more to add, you think? Or are we getting to a good coverage of...
So there are two kind of sides to that question again. One is whether, we are catching everything that's known, and I think that we can tackle by working together to catch all the known receptor-ligand interactions. But another side to that question is that, the protein biochemistry, biophysics, structural biology, genetics community has not mapped all the interactions, and there are still so-called orphan receptors we don't know what the partner is, so they're orphans, right? And so the question is, and something that we're working on is, can we use this data, but also things like AlphaFold, to inform on what are those orphans, who are they talking to actually, and the ones on the cell surface, what are they seeing on the other cells, and can we actually harness both the human cell atlas and protein structure prediction to finish the puzzle on a cell extrinsic point of view. And of course there's also the same thing is cell intrinsically to some degree.
More questions?
Hi, thank you so much. So a few years back, there was a paper in Cell showing that there are some different thymic epithelial cell populations that deterministically express lineage-defining transcription factors.
Which ones?
A Cell paper, I don't remember the authors.
Okay, that express the...
Yeah, the thymic epithelial cell populations that do not express ID,
okay.
They deterministically express.
Yeah.
So do you know? Did you check if there's any different spatial organizations of these tag populations in your data set?
Yeah.
Thank you.
Yeah. I mean, to some extent. So what he means is they are air expressing and non-air expressing subpopulations of thymic epithelial cells, and where are they located? So, I mean, there are some populations that have clear localization, like with respect to the Hassel's corpuscles. Other populations, I mean, both air expressing and air non-expressing, at least with this resolution of 50 micron, it's not clear that they have any particular localization. So there's kind of, two different answers.
More questions? I have a question.
Yeah.
you see these two kind of special trajectories for CD4 and CD8.
Yeah.
And do you see a change on the transcriptome for CD4 and CD8 as they go along these trajectories?
Yes.
And do you think...
So both a change in the transcriptome and a change in the antigen receptor usage. And
exactly. Yeah,
we do the
that was
trajectory
going to be
inference
my second question.
with the dandelion. So it's integrating both the VDJ segments of the TCR alpha beta receptor usage and the transcriptome. And that gives us the power to actually see this sort of divergence much earlier than, well, not much earlier, but a bit earlier than was possible before.
Exactly. So the point that you see it much earlier, I guess, are you thinking about using this information for the in vitro kind of modeling?
Yes, yes, exactly.
If you find it from the very early, so you have some factors.
Yeah, great minds think alike. Yeah, that's what we're. That's it. Yeah.
More questions? Yes, please.
Hi. So regarding the human cell atlas, for now it's really about individual organs and how they develop. But do you see this developing into a population-level atlas where you see differences between individuals, going a bit further from differences between organs or developmental stages?
Yes. So I see the human cell atlas as sort of providing a healthy reference, of male, female, the different stages of life and so on. And then the population, we're already capturing samples from tens of thousands of individuals. So the human cell atlas is different from the Human Genome Project in that the samples are taken from many more individuals. Of course, creating like a genetic, global human variation cell atlas and exposome kind of cell atlas, that's, I would say, doing that in a more comprehensive way, that's like the next phase after we have a rough. It's sort of like GWAS to Human Genome Project, and that is important. And it's already starting for blood, for instance, or for things that are easily accessible. I think it will take more time for like internal organs and things that are harder to access, tissues that are harder to access.
So thank you so much. It was a
Thank you.
fantastic way to conclude the meeting.